So it's now my pleasure to introduce our first speaker today, Dr. Augustine Lujan. Dr. Lujan is a physician scientist dedicated to advancing research in pediatric rare diseases, including conditions like TANGO2 Deficiency Disorder. He holds a PhD in biological sciences and a postdoctoral training in cell molecular biology. At Hospital St. Joan de Dieu, he works as a researcher in the neuromuscular and arrhythmia sudden death units, serving as a bridge between basic and clinical research in this field. In addition, Dr. Lujan serves as a TANGO2 Research Foundation Research Committee member. Please join me in welcoming Dr. Augustine Lujan.
Can you listen to me? Okay. Well, first of all, thanks for the presentation, and thank you to the organizations to invite me and give me this possibility to present our science. Today, the idea is to present what I did during my postdoc in the Center for Genomic Regulation in Barcelona, and it was supervised by Dr. Vivek Malhotra. So, first of all, I have to say that I'm a member of the Scientific Research Committee of the TANGO2 Research Foundation. And the idea of this talk, this 2-hour talk, no, I'm joking, it's only 30 minutes. It's to tell you, to tell us, or you, that what we were doing during my postdoc and what we are doing now, that I moved to another place, and I will talk about that later. So when I started this project, there was background information about TANGO2. As you many know, TANGO proteins were described by Dr. Malhotra in 2006 and were published in Nature by this postdoc, Frederick Bart. And the idea of this project was to understand what were the components of the secretory pathway that doesn't have any function before. So what they did was something very simple but very clear, that was to remove different unknown genes and see what happened in the cells when you don't have those genes. So the outcomes that they were looking was proteins, alterations in protein secretions, or also alterations in the organization of the Golgi. That's why they came up with these 14 genes with unknown names. That's why they put TANGO, because it means transport and Golgi organization proteins. So proteins that when you don't have it, you can see in a cell a problem at this level, at the protein secretion or at the Golgi organization. So most of the work of the lab was on TANGO1, that is a protein that is related with the collagen secretion, and it's another totally different gene than TANGO2, and all the TANGO genes are totally different in different chromosomes with different functions. So after that, what I read first was the paper of Dr. Lalani, in which they show for the first time that when you don't have TANGO2 in humans, you can find a disorder that they explain very well in this paper. But what was important for me was that they show that there is an increase in the ER stress when you don't have TANGO2 in the system, but also there was a reduction in the volume of the Golgi. So there was some problem at the ER Golgi network. At the same time, the group of the paper of Kremer in Germany, they show again the phenotype in humans when you don't have TANGO2, but also they put for the first time, to my knowledge, for the first time, that when you don't have TANGO2, there is a functional defect in the mitochondrial beta oxidation. So there was something related with the metabolism too. So then genomes show that the subcellular localization of TANGO2 was primarily in the cytoplasm. And it was the first time that I read that the TANGO2 has more than one isoform. So here they show the location of TANGO2 isoform 1, 4, and 5. But all of them were in the cytoplasm for them. So then I read the paper of Michael Sacher, and they showed that the problem was not that the TANGO2 was in the cytoplasm. The problem was the place in which you put the tag in the protein. So if you put the tag in the N-terminus, all the proteins appears in the cytoplasm. And this is because the N-terminus of the protein is the functional part of the protein. So you are modifying the function of the protein if you put the tag there. But if you put the tag on the C-terminus, as here, you can see that there is an increased colocalization between TANGO2 and the mitochondria. But something for me very important of this paper is that they show that only if you take the first 40 amino acids of TANGO2 and you put the tag, you can see that the localization of this small plasmid is the mitochondria. So in the first 40 amino acids, it's the mitochondrial targeting that the protein has to go there and do the function in this compartment. So more than that, they show that there was a delay in the movement of cargos between the ER and the Golgi, and also that the protein was colocalized in the mitochondria by the N-terminus part of the protein. So then the group of Dr. Lina Galul-Gonzalez detailed very well that the problems in the metabolism show that when you don't have TANGO2, there is an increase in superoxide production, but also there is a decrease in beta oxidation and ATP levels. So again, we start to know that there was a problem in the metabolism when you don't have TANGO2. And also, they show that if you extract mitochondria and do Western blot, you can see that there is an increase or there is a presence of TANGO2 in those mitochondria. So there's the second time that we see that TANGO2 could be in the mitochondria. So this was the context in which we start to work in this project. And the first thing that we did was to colocalize the protein as our colleagues. And we can see that the isoform 1 of TANGO2 is colocalizing very well with mitotrackers or with the mitochondria. And as you can see here, you can see in yellow the merge between the red channel and the green channel. So that means when you see yellow, that both markers are in the same time at the same place when you take the photo in the microscope. But to be sure, we did a different approach that is called FRET. It's an approach by microscopy in which you observe how far or how close are two proteins in a system. So you take the protein A, you make an excitation with a laser, and you keep the emission of the second protein. If you see a positive FRET efficiency like we observe here, means that these proteins are closer than 10 nanometers. So for some people, this means that are interacting. For other people, says that just are closer than 10 nanometers, and that's it. But this super well colocalization between the mitochondria and TANGO2 start to make some ideas to us, and we made a plasmid in which we express a small part of the TOM20, that is a protein that is in the outer membrane of the mitochondria, with a GFP tracker, and we co-transfect the cells with TANGO2. And what we can observe here, this is the mitochondrial marker in green that is in the outer membrane of the mitochondria. The mitochondria has two different membranes, one outside and one inside. And you can see the outer membrane in green and TANGO2 inside. As you can see here, this is only TOM20, and this is TANGO2 inside the mitochondria. So if you do a video, you can observe that always the TANGO2 is inside this mitochondria. But to be sure that this was true, we did a very easy but clean experiment that is using proteases, in which if you add proteases to a compartment that has a membrane, you can cut all the proteins that are outside, but you don't modify the proteins that are inside. But then when you add a detergent like TX100, you break all of this. If you take these three samples and load Western blot, you can observe that proteins that are in the outer membrane of the mitochondria, when you add proteinase case, are cleaved, so you don't have any more. But proteins that are inside the mitochondria in the inner membrane are not cleaved, appear there. And TANGO2 has the same pattern as proteins that were in the inner membrane of the mitochondria, or proteins that are inside the mitochondria. Also, it's clear that there is a huge reduction between the first sample and the second. So there is part of TANGO2 inside the mitochondria, but there is part that is also outside. So with this idea of the colocalizations and that TANGO2 could be inside the mitochondria, we start to see the isoforms of TANGO2. There are 6 isoforms described in UniProt, but some people are looking for other options of isoforms. We just be focused on these isoforms that are in UniProt, and we choose the one that is more identical to the isoform 1, that is isoform 2, and the one that is less identical or the most different, that was isoform 5. So if you remember that Dr. Sacher says that in the first 40 amino acids, there was something important to colocalize or to localize the protein in the mitochondria, we start to analyze these first 40 or 60 amino acids in our case. And we observed that isoform 2 and isoform 1 has the same amino acids in the first 60 amino acids, yes. But isoform 5 was totally different, lacked a very big part of these amino acids that were in the first 60 amino acids. So we did different plasmids. This is isoform 1, as I showed you before. If you colocalize with Mitotracker, you see with Mitotracker, there is a colocalization between 2 proteins. If you see isoform 2 is the same colocalization as isoform 1 that you can see in the mitochondria. But if you observe what happened in isoform 5 is that it is changing the localization. It's not anymore in the mitochondria, and you see it in the cytoplasm. And we also observed these small dots of proteins that were very weird for us. So we decided to analyze what is happening in these first 40 amino acids of the protein. And we find we did an alignment to observe all the TANGO2 in different orthologs in different species. And we observed in these 4 amino acids, first amino acids, that there are 2 different domains that are very conserved. One, we call it LIL, and the second, we call it NRDE. So we try to modify those motifs and see what happened in the cell when you modify them. So as you can see here, if you've modified the NRDE domain, you see that the localization is still the same. It's the same as in the mitochondria, but you can look that the mitochondria looks different. It's like more fissionated or something is happening at that level of the mitochondria. But if you make a mutation only in the LIL domain here, you can see that the localization is totally lost. So now you see that the protein is outside, it's in the cytoplasm, and it's making like weird blobs of proteins, like aggregations of proteins. Here we have a body pie. Body pie is a staining that we use to see lipid droplets, so neutral lipids in the cell. And as you can see here, when you have these blobs of the proteins that are in pink, you can see that lipid droplets are close to these aggregates of proteins, like in contact. And there are many of them. So if we remember that the first 40 amino acids were important for the localization, and then we thought that the LIL domain is the real part of the protein that is important for the localization, we make a mutation to see the first 40 amino acids, the normal ones, and we can see that the localization was in the mitochondria, as Dr. Sacher says. But when you remove only the LIL domain in this first 40 amino acid, now you lose all the localization of the protein. So we can say now that the LIL domain is very important for the localization of TANGO2 in the mitochondria. But then I show you that when you mutate this LIL domain, this protein makes aggregates and goes outside the mitochondria and contact lipid droplets. So if we express TANGO2 isoform 1 with GPA-T4, that it's a small protein that appears in lipid droplets, you can see that are very close in some parts in which mitochondria is contacting lipid droplets. This is something super well known. But if you express the TANGO2 LIL, in which the protein goes from the mitochondria to the cytoplasms and contact lipid droplets, you can observe that now you have a lipid droplet in green here and protein of TANGO2 that is in the how do you say? It's not a membrane, in the monolayer of the lipid droplet. So it's contacting the lipid droplets. So there is something in these lipid droplets that allows TANGO2 to contact them. And there are many. If you see, there are many dots that are colocalizing. So after that, we start to study the structure and the pockets that has this protein, as many groups did. And we use different approaches and we use different softwares to describe the pockets that we can find in the protein. So we use a KB Finder, DeepSight, and P2 ranks to see those pockets. And you can see here in this table is all the pockets that are detected by a KB Finder. But for example, DeepSight only describe or recognize 4 of them, and P2 rank describe only 3 or recognize only 3. But for us, the most important was this one here that we call it F. And it's because it's a huge pocket in the middle of a protein that it's very globular. So it's like a C, the protein. So as you remember, in our first publication, we saw that when we removed TANGO2 from hepatic cells, what we can observe was that there was an increase in lipid droplets, in the accumulation of lipids in these lipid droplets, in numbers and also in quantities, depending on the cell type that we use. This is a representation of normal cells with lipid tox, is staining for lipid droplets. And this is when you don't have TANGO2. So there is an increase in this accumulation of lipids. Then we made an analysis to understand by lipidomics the quantities and the qualities of these lipids, and also all the enzymes that are involved in the lipid metabolisms to understand what was happening at the level of the cell. But something that was very important that described Dr. Lina Galú-González was that there was an increase in the superoxide inside the cells when you don't have TANGO2. And for us, it was important to understand if this amount of ROS was capable to modify the lipids. So what we did was to check that was true that there was an increase in ROS when you don't have TANGO2, but also to check the lipid peroxidation. So if this ROS could modify lipids, because when you modify these lipids and you oxidize them, then you cannot use it anymore to produce ATP. So it's now described like 2 years ago that when you have this lipid peroxidation inside the cell, one of the mechanisms that the cells use is to put it inside the lipid droplets because these lipids could be toxic for the cell and could induce apoptosis. So after this first idea of this paper, what we observed was that there was an increase in lysophospholipids and a decrease in phospholipids. And then Dr. Saher and Dr. Vandana Gupta made different approaches using lipidomics too, and they observed similar results in terms of lysophospholipids, phospholipids, and an increase in the accumulation of neutral lipids. So with this idea, we start to think another hypothesis, and is that to produce a phospholipid, you need to take a lysophospholipid and add a acyl chain. This is called acylation, and these mechanisms is done by an enzyme that could, in this case, for example, lysophosphatidic acid, you need the lysophosphatidic acid acyl transferase. It's a protein that transfer this acyl chain and produce the phosphatidic acid. And this is for all the lysophospholipids, or most of them. So to do that, you need an acyl-CoA, and then you put this chain here and remove the CoA. So there were two main questions to answer here. First of all, if it is TANGO2 possible to do the work of an acyl transferase, and second, if TANGO2 is not an acyl transferase but could be an acyl carrier that gives the acyl-CoA to the transferases to do the job. So with this idea, we start to analyze all the acyl transferases that are known, are all these here. And as you can see, most of them share motif 1, 2, 3, and 4 are different motifs that appear in this population of acyl transferases. And there is something interesting because all of them has a transmembrane domain. So our protein that has to be attached to a membrane. But the only one that doesn't have a transmembrane domain is this one here. CGI is one of the latest that were described and has this motif 1, and just with that could have activity as an acyl transferase. So we analyzed the structure of TANGO2, And we observe that there is a region that could be the motif 1 of the protein, if it is an acetyltransferase. There are 2 regions capable to be the motif 2. We didn't find a motif 3, but we find a possible motif 4. So with this idea, we contact Professor Hideyoshi Indu in the University of Tokyo, because he is an expert in lipid science, but also he described some of these acetyltransferases before. So he did a very simple experiment in which, if we say that to produce a phospholipid, you need lysophospholipid, an acetyltransferase, and acyl-CoA, you can do that. You can put your protein that is or should be an acetyltransferase, you put the lysophospholipid, and you put the acyl-CoA. So if this works as an acetyltransferase, then you will see after a few minutes or time that appears the phospholipid, because you are producing it. So they tried with many different lysophospholipids, and they didn't find any acetyltransferase activity in TANGO2, at least using our approach. So with that result, we say, okay, maybe it's not an acetyltransferase, it's the protein that is giving the acyl-CoAs to the acetyltransferases. So we did a very clear experiment that is simple. It's using a fluorescence quenching assay, in which you have a lipid that has coenzyme A in one part and has a tag that is fluorescent in the other part. So the idea of this is, first of all, you can make soluble a molecule that is hydrophobic, if not, and then you can see it because you have a fluorescent tag. So if your protein interacts with this component that has a fluorescence, the interaction could make a drop in the fluorescence intensity, because you have the fluorescence intensity, then you have a protein that is attaching this molecule, and you cannot see anymore the fluorescence because you are keeping that tag inside. So we did that. We add the MBD 16-carbon CoA, and we add our protein. As you can see here, this is the lipid that you buy in the market, and this is the fluorescence that has. When you add your protein, the fluorescence drops because you are making a contact. And to be sure that this contact is true, then you add a competitor as Twin 20, that is a soap that could break that interaction. So you can see that you recover again the fluorescence that you have before. So after that, we say, okay, if TANGO2 has this domain called NRDE that is inside the pocket, that is important for the function, what happens if we mutate that domain? So we did that, we mutate the domain, and we do the same experiment. And as you can see here, this is the fluorescence of the lipid. When you add the protein with the mutation, now you don't have this big drop that you have in the other situation. So at least in this, with this approach, we start to understand that the NRDE was important for the contact between TANGO2 and the lipid. But during, when we sent this paper, some reviewer, and I really have to say thank you, because recommend to do a competition assay. And this for me was amazing, because it was something that gave a lot of more information and more strong results for us. And the idea is, it's also simple, in which if you say that when you put the protein to the lipid, you have the drop, what happens if you put more lipids alone without the fluorescence? Now the protein has to compete. There is a competition between the lipid that doesn't have the fluorescence and the lipid that has the fluorescence. So the protein cannot choose and will attach one or the other. So now the fluorescence has to be less. And as you can see here, here. If you add more lipids without the fluorescence, what you can see is that now the drop is less, because there are parts of the protein that is attaching to this lipid that doesn't have the fluorescence. So we try the competence with other lipids to see if TANGO2 has a preference for some kind of lipids and not to others. And what we observe is that if you put a longer lipid instead of 16-carbon 18 and 1 in saturation, there is a less drop. So means that TANGO2 prefer this lipid instead of this. And finally, to be sure that the protein was not attaching the CoA instead of the lipid, we add CoA alone. And what we observe is that if you add CoA, it's the same as the previous result. So if you add CoA, there is no difference in the competition. So it's not attaching the CoA, it's attaching only the lipid part of the component. So with this, we start to think that the NRD was important to contact lipids, because when you don't have it, there is a break in this contact. But when we produce the protein, something very weird happened, and was that we didn't find the protein alone. When we produce the human protein in a human model, what we observed is that the protein was making oligomers. So the protein was not only was not alone, was attaching other proteins that are same as TANGO2. And when I did my PhD, I worked with proteins that do that, like galectin-1 are proteins that are in a balance in nature between monomeric and dimeric conformation, and that it's very important to produce the function. So in this case, when we observe this, we try to think if this was true or not. And we did an experiment that you take a protein, a plasmid with your protein, TANGO2, you put a flag that is a tag, and you take another protein that has another tag that is called RFP. So if I use a bead with anti-flag, I will pull down this protein here, and if there is a true interaction between these two proteins, I will see this fluorophore after. And we do that, and we observe that. So if you pull down the TANGO2 with the flag, with the anti-flag bead, you can recover anti-RFP, so RFP. So it's true that there is interactions between TANGO2 by itself. So finally, we came with this model in which we can see that if you force the in silico model, you can see this is TANGO2, the NRDE domain is inside the pocket, and you can put an acetyl-CoA. And if you force the system to put more and more lipids, the system is capable only to add 2 different chains of lipids inside the protein, because if you put 3 or 4 or more, now the lipids appear outside the protein, and it's totally weird. So finally, I want to make a summary of this information, this presentation, in which TANGO2 for us is localized within the mitochondrial lumen, contains a molecular pocket that could accommodate acetyl-CoA. The LIL domain is important for the localizations in mitochondria. The NRD domain is very important for the interaction with the lipid components. And TANGO2 can form oligomers. So what we are doing now, as many other groups, biomarkers. And why? Because we took the secretome of fibroblasts from patients, we ran a gel, and we observed that there is a huge difference between the secretion of TANGO2 fibroblasts compared to healthy donors. So with that, we received recently a grant from the TANGO2 Research Foundation, and we will do a collaboration with the CRG and Hospital St. John de Deu to check if what kind of proteins are this one, and if we can use it as a biomarker. But finally, I think that the most important part of this presentation is not the data, it's just my open questions, because I have many questions. I have more questions than results and more questions than answers. And I think that the most important part of these presentations and these days that we are sharing is to share also our questions and our ways to resolve these problems. So these are parts of my questions, but I have many, many others. So everyone that wants to talk about it, please stop me somewhere and we can talk about. But if there is an increase in ROS production, and we know that there is a dysfunction in mitochondrial, why we cannot think that maybe antioxidants could be a very good strategy to improve our treatment. So also, I don't know which is the mechanism in which TANGO2 could be inside the mitochondria, because the protein that goes inside the mitochondria needs to be unfolded, entering the mitochondria, and then are folded again. So this is something that is important to understand if this is true. Then if TANGO2 has 6 different isoforms, what are the functions of all of them, and where are in the body? So all of them are equally important, or we have to concentrate just in those that appears in the nervous system, in the heart, in the muscles. So we have to think about it. Think if TANGO2, the oligomerization of TANGO2 could influence the biological function as do in galectins. And also if TANGO2 has this capacity to transfer lipids, could be or not a protein similar to those that appears in the membrane of the mitochondria, in the inner membrane, taking phospholipids from one membrane and put it in the other. And these dynamic functions allows the mitochondria to do their job. If you need to produce energy in a mitochondria, you need that there is a dynamic membrane that could produce the fission and fusion of the mitochondria. So just to finish, time is up. To my knowledge, this is an African proverb. I think I totally agree with this sentence, in which if you want to go fast, go alone, but if you want to go far, go together. Finally, I want to thank Dr. Vivian Malhotra and Umberta Foresti, that were the people more involved in this project, all my colleagues, all the people that make collaboration with us, all the people that give the money to do it possible, and thanks. This is all.
Thank you so much, Dr. Lujan. So we're going to have audience Q&A. If you have a question for Dr. Lujan, I'll ask that you stand and I'll come to you with the microphone.
Thank you, Agu. So more questions than answers.
Always.
When you say TANGO2 is in the mitochondrial lumen, where in the lumen? Is it the matrix, the inner membrane? Is it in the intermembrane space? Does it make a difference?
Yeah, it's a great question, because we are trying to understand that. We just know that it's inside, not where. If it is in the intermembrane space, we're trying to do. There's an approach to check that, and we're trying to do it. It's not easy, because we need different plasmids that you can express proteins that are only in the intermembrane, only in the interspace, in the intermembrane, and inside the intermembrane. So we're in contact with the group that they use this kind of experiment, because I tried before I left CRG and I couldn't. So now there is a master student in the CRG working on TANGO2, and she's trying to understand that.
Nice talk. Right here. Just a quick question. In working with the acyl-CoAs, one issue you can have with those is they notoriously form micelles. So the labeled in with the fluorophore could certainly be. How do you combat that during these measurements, or did you find that to be an issue?
You mean the fluorophore in the chain, or
yeah? Because acyl-CoAs can form micelles at low concentrations and could certainly affect the measurement.
So to do that, we contact Eric Supen, that it's a researcher here in US, that he described the same approach that I show you. He described it, and I had a talk with him to understand how to do it, and we followed that protocol. We had some controls to understand that this was working or not, that I follow all these steps that he told us. I really don't know if there were missiles or not. What I understand is that these two different components that we buy in the market, when you put it on liquid, start to be soluble. So don't produce these missiles, because it has the CoA and the fluorophore that has something happen with the fluorophore there. But I'm really not an expert on that. We can discuss it, and I can show you all what I did. So
Great, thanks.
super good to understand. Yeah.
Hi, good afternoon. I joined completely late. I'm Cindy Garcia. I'm a parent. I do have a question. I'm not very well versed with everything that's going on, of course. But for your first question, it really kind of stuck to me, the antioxidant-based therapeutic strategies. Do you think maybe glutathione could be one of them because it is used to combat cellular damage and oxidative stress?
So I put this question because I think that it's something that I want to discuss with the clinicians, because here there are many clinicians, and even I'm a medical doctor, but I don't see patients. So I don't want to say we have to do this or that. I think that this is something that is going on the medical doctors. So I put that question because I have it like you, and I put the pressure on them. But just to discuss if this is possible or not, and if we have risk or not, but just an open question. It's difficult to say it in my position because I see it in cells. So in cells it's easy because I can put antioxidants and I see something better, it's okay. But then it's different to go and say it to the patients. So I want to discuss with them. We can do it during these days, and let's see if at the end we could have a more clear idea. You're welcome.
Excellent talk. For one of your questions, have you tried to map known patient mutations to different transcripts and then try to look at what is the phenotype of these patients, isoform 1 versus isoform 6 versus isoform 2?
Not at the level of isoforms. What we tried was to, there are some mutations describing patients that are affecting only the LIL domain and the NRD domain. We did the same in the lab. We observed same approaches and the same results as when we remove or mutate all the LIL and all the NRD. But could be also important to see what happened with the isoforms. Also, I think that it's necessary to see the expressions in all the tissues, making in one way.
Agu, for the secretome experiments that you are going to do, or probably already are doing, are you using any other disease controls? And the reason I ask is you could take a VLCAD patient, run their cells, and then subtract out that kind of non-specific background from what you see, maybe hone in on something that's more specific to TANGO2.
We didn't put it in this project, but could be a good idea to add it. It's not easy. We have patients with this other disorders, so we can try to see if something is there. Now our discussion is, if we want to do it, there's a discussion between, okay, we collect the secretome, and the secretome has many things. In those many things, there are also exosomes. I'm a little bit passionate with the exosomes because now there are more evidence that, for example, if you have a mother that it's before the birth, you can take exosome from the kid that is inside in the blood of the mother. So if we can find these biomarkers in exosomes, then I want to try if I can check as an early biomarker in these situations. But it's very difficult to do this. I never think about to add another disorder, but we can try.